My name is Adelie Castillo, and today I'll be talking about the synaptic transmission. To begin, in the nervous system, a synapse is a structure that allows a neuron to pass an electrical or chemical signal to another cell, and then we have the synaptic cleft, or the plasma membranes of each neuron in very close contact separated by a narrow space, and the presynaptic cell, or the sending neuron, delivers the signal to the synapse, while the postsynaptic cell, or receiving neuron, receives the signal once it crosses the synapse, and these neurotransmitters are stored inside vesicles found in the presynaptic neuron. We will be focusing on the chemical synaptic transmission, which comprises most of the synapses on our body. We know that the synaptic cleft separates the pre- and postsynaptic cell, and then we have this action potential coming from the axon terminal, results in the secretion of these neurotransmitters from the axon terminals, and the neurotransmitter molecules diffuse across the synaptic cleft and bind to the receptor proteins on the cell membrane of the postsynaptic cell, and this binding of the neurotransmitters to the receptors on the postsynaptic cell leads to this transient change in the postsynaptic cell's membrane potential. And right here we have a diagram which shows the steps of an action potential occurring from this depolarization. We have, when this action potential arrives at the axon terminal, it activates these voltage-gated calcium ions in the cell membrane. The calcium, which is present in much higher concentration outside the neuron than inside, rushes into the cell. The calcium allows synaptic vesicles to fuse with the axon terminal membrane, releasing neurotransmitter into the synaptic cleft. And we know that the molecules of the neurotransmitter diffuse across the synaptic cleft and bind to the receptor proteins on the postsynaptic cell. And this activation leads to the opening or closing of these ion channels in the cell membrane, which can produce the depolarizing or make the inside of the cell more positive, or hyperpolarize, make the inside of the cell more negative, depending on the ions that are involved. And we know that these changes in chemically gated ion channels create these synaptic potentials, which enable one neuron to affect another. There's two types: the excitatory postsynaptic potential, EPSP. A neurotransmitter binds to this postsynaptic receptor and opens sodium channels. The ions diffuse inward, which causes a depolarization in the membrane, possibly triggering an action potential. It makes the inside of the cell more positive. And then we have the inhibitory postsynaptic potential, IPSP. If a different neurotransmitter binds, it increases membrane permeability to potassium ions, and these ions diffuse outward, which hyperpolarize the membrane. We have this less likely to fire an action potential. We'll be talking about these neurotransmitters. Their nervous system produces at least 100 different types of neurotransmitter in the brain alone. We know that some neurons release only one type, whereas others, they can produce 2 or 3, and these different types, one of them is acetylcholine, which we know stimulates skeletal muscle contraction. Another group is monoamines, a group of modified amino acids, which includes epinephrine, norepinephrine, dopamine, and serotonin. Another group is unmodified amino acids: glycine, glutamic acid, aspartic acid, and GABA. And we have the last group, which are peptides, which are short chains of amino acids, which include encophalins and substance P. And we know that there's also 2 different types of neurotransmitters, which are excitatory, which increase the postsynaptic and membrane permeability to sodium ions, bringing the postsynaptic membrane closer to the threshold and may trigger the impulses, and inhibitory, which we see that this reaching threshold is less likely. They decrease the change, and the chance an impulse will occur is less. And we know that to prevent the postsynaptic cell from continuous stimulation, we need to distort or remove the neurotransmitter from the synaptic cleft, which we have 2 types of removal: reuptake and enzyme degradation. Reuptake, the neurotransmitter is reabsorbed back into the presynaptic neuron and restored inside a vesicle ready to be used again, for example, noradrenaline. And enzyme degradation, the neurotransmitter is broken down by an enzyme into a smaller inactive product, which are then reabsorbed by the presynaptic neuron and resynthesized into active neurotransmitter. And we know that we have this diagram which shows these 2 different mechanisms. We see reuptake and the deactivating the enzymes. And these are my resources.